Thank you, Kieran, for the nice introduction. Also, thank you for the perfect organization of this school. Kieran has done an excellent job. He has, by himself, put together this entire program and including the social activities. So, thank you very much for that. I also had the occasion to welcome those of you who traveled to come here because I was not here yesterday to do that. So, I take the occasion to give you a personal welcome to Imperial College London, Department of Bioengineering. As you know, this is the URM building dedicated to engineering and medicine, and we are currently at the 12th floor. The main activities related to this school are located in the 4th floor, actually. And just to give you a bit of perspective, in the 4th floor of this building, and those of you who stays a bit longer may visit it are all related to human interfacing and augmentation. These are the PIs in this floor downstairs. You actually have heard from Etienne Bourdet yesterday. You will hear from Iri today, actually. You will also hear from Juan Gallego. You will hear from me. So you will hear from most of the people in this floor. Which is dedicated entirely to human interfacing and augmentation. My group, as Kieran has anticipated, is mainly focused on neuromechanics research in a broad sense, from, well, almost we can say from animals to full clinical applications, and the development of rehabilitation technologies. And this is a snapshot of a number of figures from our papers to give you a glimpse of what we do. Today, I will talk about some, at least for the first part, those of you who have listened to one of my lectures before, they will find some repetition. There will also be some topics that you will hear again during the week, I'm sure. And then the second part, though, I put something that is a bit related to the newest research. So it's a bit of a balance between being didactic at the beginning, for those of you who are not familiar with these topics, and then going a bit more deeper and towards the most recent findings in a few examples towards the end, so that those of you who are more familiar with these topics will also find something interesting today. So in our group, we work on movement and everything that concern physiological and pathological movement, including methods to restore impaired movement in patients. Movement sometimes is underscored the fact that it's the only way in which the brain communicates with external worlds. Of course, I'm now talking to you. I'm using the movement of my facial muscles and my vocal muscles in order to communicate with you. Without movement, the brain cannot communicate with the external environment. And actually, the brain evolved in animals with the primary aim, or the only aim actually, of moving. All the rest can be considered a byproduct. So that means that understanding brain function cannot be done without studying and understanding the movement, which is what the brain has evolved for. When we talk about movement, there is, in terms of research in movement, in terms of studies, the so-called neuromechanics of movement, there is a well-known gap between different levels of analysis that corresponds also historically to different disciplines. On the one hand, we have the study of movement through the forces that are exchanged between the musculoskeletal segments, and that the behavioral analysis or biomechanical analysis, if you want. And that is a very established field. We have very detailed models to describe movement at this level in a very precise way. And then on the other hand, when we talk about neuromechanics, so you have the word neuro and the word mechanics. So this is mechanics, but this is the neural part. And this is the neuroscience, the neurophysiologist, who are interested in going deeper and deeper into the cellular, even subcellular mechanism of movement. And of course, the deeper you go at this level, the farther you are from the behavioral level. So one of the main limitations in understanding human movement, as you can see in the sentence reported, that is our poor ability to record and decode from a sufficiently large number of neural cells in order to be able to associate the functional activity at this level, which is the neural level, with behavior. And just for those of you who are starting their career, I have this slide since more than 20 years. I use this slide when I had to prepare my inaugural lecture for my first professorship, and I didn't know exactly what to put as a long-term vision. This was 20 years ago, actually more than 20 years ago, in Denmark, in Aalborg, and I had exactly this slide. I had not touched it at all. It's 20 years old. So that also indicates that sometimes you may have a problem, challenge, and you may keep it for your entire career, maybe without solving it, but at least to addressing some of its parts, as we will see today. So this is the fundamental limit in neuromechanics, and also the fundamental challenge, but also the exciting part in neuromechanics is try to fill in this gap, right? And if you think at that, this is exactly the same problem that you have when you develop a neural interface, right? When you develop a neural interface, you have exactly to do that. You have to associate to the neural activity that you try to decode, you have to associate a function in order to control neuroprosthesis, which is exactly what we have said in the previous slide. So neuromechanics research and neurointerfacing research are fully overlapped. They have the same challenges. If we can solve challenges in one domain, we are solving challenges in the other domain, and vice versa. And indeed, neural interfacing advances have been very useful in the past decades to solve a lot of motor control and neuromechanics problems, and of course vice versa. Right. In order to again at a more didactic level, in order to give you an example, a simple example of the gap that we are discussing, so the gap between the neural and the functional part of movement, I will just provide you a simple example. The simple example starts in this way and lasts a few slides. We'll understand in a moment where I'm going to. So this is an example of motion synthesis with a reinforcement learning algorithm. This is work of Balint Todossi here. And this is an agent. In this agent, there are all the rules of biomechanics, so the agent will collapse unless you can program specific torques at each of the joints, which is what the reinforcement learning algorithm does with a number of training trials with a reward in order to have a locomotion that mimic, at least partly, the real human locomotion. You see, maybe the steps are a bit awkward in some cases, but that is not that far from real locomotion. We can complicate this. We can also ask the agent to do things that are a bit more complicated, like having steps, and he has to adapt. You can even just out of curiosity, I show you this, invent very complex tasks that we will not have in natural conditions. But why am I showing you these videos? It's just because this is a sort of behavioral level, right? So in this case, we have somehow with a reinforcement in the learning algorithm understood through the algorithm the behavior, and we can replicate the behavior in complex situations. We can replicate almost perfectly human behavior. So if we can replicate it, it means we understand it, right? We can look at the individual forces in each joint. We can understand how these forces are coordinated in order to do this task and many other tasks. All right, so that's the behavioral part. And in this simple example, you can replicate it. So now let's ask ourselves the simplest question that we can ask from the neural part. And the simplest question is a question that has afflicted biomechanicists for decades, which is the following. Given the joint torque that we have computed before, so before we know all the joint torques, right? How does the central nervous system produce this torque, given that there are a number of muscles around this joint, given that there are a number of muscle compartments also in that joint, ultimately that there are a number of muscle fibers, each of which is producing a certain biomechanical action. So, as I said, we are at the very basic neural side, But yet, if you ask yourself this question, you cannot answer this question unless you do something more that we will see in a moment. And why cannot you answer this question? Well, it's obvious. All of you knows that this is an ill-posed problem because you have one equation, but you have a number of uncertainties, specifically the activation of every single muscle or even every single group of muscle fibers, right? Every muscle compartments. So how do you solve the problem in biomechanics? Well, if you only look at the biomechanical side, you say, let's put constraints. Let's put the constraint, for example, that those muscles cannot be active for a very large amount. So let's minimize the maximum activation among all those muscles. You can put a number of other constraints. As soon as you put enough constraints, then you can solve the problem, and then you have solutions. And the solutions will depend on the constraint that you put. So these are examples of solutions. The colors are not perfectly clear, but you will see that the actual activation of the muscles is very different across these solutions, and those are related to the various ways in which you can solve that equation. Of course, what is the correct one? Well, maybe none of them, right? Especially if you then consider a pathological condition, certainly none of them is the correct one. And so what is our conclusion? That the very simplest neural level that we do after having reproduced movement in such a reasonable way as we've seen before does not even answer. And of course, there are much deeper neural questions, right, that you can pose yourself. So this is the simplest possible. So what do you need to answer this type of questions at the neural level? Well, you have to interface. You have to do what is called, as we mentioned before, neural interfacing and also decoding. Interfacing means that you are recording from neural structures. Decoding means that you are extracting information from those recordings, right? And you can interface at various levels of the neuromuscular system. You can put electrodes inside the brain, inside the spinal cord, in peripheral nerves, in muscles, and so on. And each of these interfacing will give you some information about the specific question you are trying to ask from the neural side. What I will focus today, as you've seen from the title, is interfacing very particular structures here, which are the very last neural cells in the spinal cord, which are also the very last neural cells in the nervous system, which are the motor neurons, the spinal motor neurons. Spinal motor neurons are located in the spinal cord. Their axons, instead of going back to another neuron, as for the other neurons in the nervous system, they connect to muscle fibers. So they are the output of the nervous system. So you can figure the nervous system, if you want to have a huge simplification, so brain, spinal cord, and all those extremely complex systems, as a giant neural network, right, which we don't fully understand, as we know, of which the output is a layer of thousands of these cells that are eventually connected to muscles. So this is the output of the system. This system is very complex. Maybe if we can read the output, we can understand the system a bit better. So that's the kind of philosophy. Being the output of the system, they had to receive input from the entire system. Indeed, motor neurons have been classically defined at the beginning of the last century by Sherrington, a Nobel Prize for Physiology, as the final common pathway of the neuromuscular system. You can see here in this scheme by Gandivia what that means. That's the spinal motor neuron. It receives synaptic input from the entire nervous system. So that's the meaning of a final common pathway. Everything eventually has to go to motor neurons, because everything eventually has to be translated into behavior. And to be translated into behavior, you have to pass through the motor neurons that are connected to muscles, right? So they're quite interesting cells because they are very close to function. So you remember the gap I was saying between function and neural structures. Now you have these cells that are very close to function, and these cells contain the very last neural code for movement. The neural code for movement is processed by all these systems eventually, as you can see, the system converges into the motor neurons, and the output of these motor neurons is effectively the ultimate neural code of movement. So it's a code that is particularly useful to understand because it's the code that is then directly transformed into function. Okay. So how many are the motor neurons? I often show this slide in my talks. So as I said, those of you who have heard a few of my talks will have seen some of these concepts few times. But it's good to remember that in a school especially, how many motor neurons do we have to perform movements? Well, we know that the cortical neurons, for example, representing the end of the arm, are in a huge number, hundreds of millions. How many are the motor neurons? How many are those at the very end? So what is the dimensionality reduction that the system is able to perform in order to still be able to determine a certain motor function? Well, this is a study that discounted the motor neurons needed to control the full arm, so the gesture that I'm doing now. And if you count those motor neurons by looking at the nerve fibers from the nerves, it is a cadaveric study with imaging techniques, you find out that on average of around 9 cadavers to be precise, is a bit more than 20,000. So you have 20,000 motor neurons to control the full arm. If you count the motor neurons that are needed to control the intrinsic muscle of the end, so not the muscles of the end that control the end in the forearm, but just those in the end. And if you think that those muscles are responsible for the most complex motor tasks in our body, these are less than 2,000. So what does that mean? It means that we don't have that many spinal motor neurons. The last neural code for movement is coded in a relatively small amount of cells. And for those of you, I think the majority who are engineers, this is a very good news, because that means that maybe we can interface every single of them. So it would be very difficult to think at a neural interface at the brain level, for example, that can interface every single neuron responsible for controlling the end, because cortical neurons, as we said, will be hundreds of millions, but it is feasible to think that maybe you can interface 2,000 motor neurons controlling the end, or even 20,000 controlling the arm, and similar for other parts of the body. So that's one good thing of interfacing motor neuron, one of the reasons why we study motor neurons. Another reason why we study motor neurons is a sort of a methodological reason, meaning that you've seen before in the slide before that motor neurons are connected to muscle fibers. So each motor neuron innervates a certain group of muscle fibers. So that means that every time a motor neuron discharges an action potential, the fibers that are innervated will also discharge action potentials, one action potential for each axon of action potential. And the action potential at the level of the muscle fiber is bigger and can be recorded with better signal-to-noise ratio than the actual action potential discharged by the motor neuron, which is in the peripheral nerve. So we can connect muscles with electrodes instead of nerves, and we can decode or try to decode the activity of motor neurons at that level. So in this case, this is something often repeated when discussing these techniques, the muscle fibers that are connected to each motor neuron, so the bundle of fibers connected to each motor neuron, are a natural biological amplifier of motor neuron activity. So we have already an amplifier of motor neuron activity. We don't need to build one. We have a biological one which is already attached to the motor neuron in a very robust way, which is the neuromuscular junction here. So that's great because then we can record the output of that amplifier. What does it then mean? We record the electrical activity of muscle fibers. So that's a very big advantage with respect to other cells for which we have to have surgery. We have to implant electrodes directly into the neural tissue. In this case, we can put electrodes not in the neural tissue, but into muscular tissue, which is very robust. It cannot be damaged. It provides a large signal-to-noise ratio, relatively simple to decode, as we will see in a moment. So, since this is a summer school, who was the first to understand that you could record motoneural activity from muscles. This Adrian, also Nobel Prize for physiology in the 30s. This paper is from the 29. He didn't get the Nobel Prize for this, for many other reasons, but this is in one of his landmark papers in the Journal of Physiology, actually a pair of papers, part 1 and part 2 by Adrian and Bronk. Bronk was one of his students. And a century ago, or almost a century ago, Adrian and Bronk proposed to insert a concentric needle into muscles in order to record the electrical activity of muscle fibers, and understood at that time that this could be directly related to the activity of the motor neurons that were innervating the muscle. And if you have time, these are very beautiful papers to read, because considering it's a century ago, it's amazing how many insights into the neural control of movement they could do from these very first human recordings with this concentric needle. And actually the recordings were only on one muscle, who was the muscle of Adrian himself, that was the triceps. But despite the very limited number of measurements, the number of insights from those papers was really impressive. Before Adrian proposed the concentric needle, motor neurons were recorded in animals directly from the nerve, and because the nerve contained, as we have seen, thousands of fibers, it was not possible to be selective. And so neurophysiologists were scratching the nerve literally with a blade until a few fibers remained. And then they could put electrodes around that almost destroyed nerve that had only 2-3 fibers, and they could record from motor nerve. So that was the technique before 29, which is also described in this paper. Of course, putting an electrode into muscle is a much easier approach. As you can see, these are classic intramuscular recordings. Every spike corresponds to a motor neuron. You see the spikes are very large. The signal to noise ratio is excellent. So a century after, we have a bit better technology for this, but not much better. The concentric needle is still used in any hospital. If you go to have an EMG examination in a clinical neurophysiology department, the doctor will examine you, will use a concentric needle identical to the one described by Adrian a century later. So definitely a technology that remained for long. But for research, we have a bit of advanced systems, and these systems, for example, these are those developed in our lab. There are other systems developed in other labs. These are filaments, so these are type of wire electrodes, in which by micromachining techniques, there are deposition of platinum that are the recording points, and then you see the tracks that goes back to a connector. So this is what we call a microelectrode array for muscle recordings. This can be inserted with a needle. So the needle can be put in this U-shaped wire. The needle is used to push the filament into the muscle. Then you can remove the needle and the filament remains into the muscle. And now, with respect to the concentric needle, you have tens of recordings at the same time. You have an array of recordings. So this is an example of 40 recordings from one of these microelectrode arrays. And so when you have 40 recordings, because you cover a larger part of the muscle, you can now identify more and more motor neurons. These are around 50 motor neurons decoded from these 40 recordings. So in the previous slide, if you have looked well, this is a classic concentric needle recording. You have maybe 2-3 type of different spikes, so 2-3 motor neurons. And now if you have a microelectrode array, you have 40, 50, sometimes more motor neurons, depending on the conditions. So remember, motor neurons are not that many. So if you start decoding 40, 50 in a muscle, that's quite a large number. If you put more on these microelectrode arrays in the same muscle, you can multiply the number. And now we arrive to a few hundred motor neurons, maybe decoded in very standardized laboratory conditions. But it's quite a nice number. You can also do that non-invasively. You can do that non-invasively by just building even simpler systems that are based that are based on, again, electrode arrays, this time placed on the surface of the skin. And in this case, this is very simple technology, of course. The electrodes are metal discs, and they have an amplifier that recorded activity. So the interesting thing is that either if you record inside the muscle with microelectrode arrays or outside, the result is that you have a number of electrical activity, which is voltages, recorded from the active muscles. So you are sampling the electric field that the muscle is generating, ultimately because of the activity of the motor neurons in the spinal cord, right? So each motor neuron discharge several times per second. There are hundreds of motor neurons. Every time there is a discharge, an electric field is generated. Therefore, there is a voltage in each point of that volume conductor, and you can sample that by placing electrodes everywhere. So that means that instead of having the classic intramuscular surface EMG, now you have a sampling that could be also quite dense of voltage, which implies of electric fields, in principle over the full limb or the full body of an individual. And these are the summed electric fields of a number of bundles of muscle fibers that are activated by a number of motor neurons, but everything is linear, right? So you know that those interference fields that are this complex sum are nothing else than eventually the linear combination of electric fields remotely generated by every single motor neuron. So every single motor neuron remotely generates on the limb a certain distribution of voltage, and each motor neuron generates a different distribution of voltage because it's connected to different fibers. So now, interestingly, we can model these biophysics in a very accurate way. So we can recently, this work from Neurodec also with us. Neurodec is a company doing EMG models. You can take an MRI of the limb and you can exactly reproduce the electric fields that are generated by muscle activity. And you can even go a step forward. If you have a finite element model that describe these biophysics, you can then take this finite element model and teach a neural network the biophysics that is here, and therefore having simulations very quick that tells you exactly what is the electric field given the neurons active, the fibers active, the conductivity of the tissues, and so on. Okay. And this is an example of this kind of simulation. So you can now have a very precise simulation of a limb. You can put as many electrodes as you want, and you can have a simulation of the response of single motor neurons. You can also even go to the upscaling level in the full biomechanics of a certain task by having a number of muscles that receive a number of inputs from motor neurons, and then you have the biomechanical characterization. Now you see the link between neuro and mechanics in these simulations with respect to the initial simulation where only behavior. Now you have this is all the neural input from motor neurons so that you can decode, for example, with the techniques that I have shown before with implanted electrodes, for example. And then you can put that in a model, and this model effectively becomes a sort of a digital twin of your system, of your neuromechanical system. And if you have a digital twin that goes from motor neurons to function and to the biophysics, then you may try to identify the parameters of the digital twin through experiments, and this is something that people in our group are doing. And this is work started by Xian Ma, who was a PhD student until recently in our group. All right, so you can do a number of things. You can invert that model. You can identify the neural activity. You can identify properties of the volume conductor. You can identify behavior. Interestingly, these, at least in a simple way, the modeling part of EMG was known since much longer, but only recently could be then really computationally solved in the way that I've shown you. So this is probably the first, actually not probably, the first mathematical model of EMG that takes into account the neural part and the mechanical part, well, the mechanical part less, but let's say neural, and you can add the mechanical part easily here, underline EMG generation. And this is Carlo De Luca. Carlo De Luca was the scientist who started the mathematical characterization of EMG. This is 78. It's quite a long time ago. At that time, EMG was studied almost as a pseudo-random signal without much of an idea of what was underlying it. And this is the model of De Luca, which is quite interesting because you have the spinal cord, you have a delta function that represent action potentials, and then you have a transfer function that is our volume conductor. That's what before you have seen with MRI as the actual limb. That is going in those impulse responses. And those impulse responses are transforming the action potential of the motor neuron into a specific action potential at the muscle fiber. And then you have that for a number of motor neurons, and the sum is what you record as EMG signal. As I said, we can now model every single of these steps in a very accurate way. We can identify a number of parameters of that model. In particular, we can identify the firing of activity of the motor neurons, and that's, remember, is the last neural code of movement, right? And we can identify them also from the mathematical point of view by noticing that this is obviously a convoluted mistus of signals. These are the delta functions that represent the neural code in the EMG that corresponds exactly to the output of the spinal cord. You can go back in various ways, including inverting very complex models from the recording of EMG back to the neural activity. And this is work of many years ago now, Vale Sholoba in Maribor, who is here with us, inverting the process for the first time in a full way, and getting, in this case, only working on statistical signal processing, the motor neuron spikings. So these techniques have been used over the last 2 decades, have been extended in various ways. You can now do it in many ways, as I said. One way is to do with a deep neural network. Another way is even to invert a full generation model, starting even from a personalized MRI of the individual as we are doing now. The interesting thing of these models is that if you look at the Luca model here, this model doesn't take into account differences between surface or intramuscular EMG. The differences between those two signals are just in the impulse responses, right? But those are parameters of the model. It doesn't matter. For the rest, that model is completely general. Actually, that model is valid even from intracortical recordings. You would have exactly the same model. If you put an array of electrodes into the motor cortex, you have exactly this generation model. Here you will not have the spinal cord, obviously, but still you have delta functions that are the firing of the upper motor neurons in this case, or upper neurons in general. You will have a transfer function or impulse response that depends on the tissues at the brain level, and then you have a multi-unit signal. So that's exactly the same model. This model actually can be extended to a number of other observations coming from neural cells. If you record ultrasound from a muscle, which is the mechanical response of motor units, you can make a model which is very similar. And or if you record magnetic fields from a muscle with magnetomyography, you have a model which is very similar, and so on. So the interesting thing is that the solution of this model applies to a number of fields with minimal changes. So these solutions can be applied to a very large number of fields. Here you see the application of these methods to a mix, for example, of surface. Here you see surface grids and microlateral arrays. Here the skin is open, but there is no surgery here. The microlateral arrays are just inserted with a needle, so you don't need surgery. And in this case, you have mixed recordings, which doesn't matter. That means that the impulse response will be very different depending on the motor neuron, depending on the recording point, but you can put it all in the same model. You can solve it all within the same solution. And this is an example I was mentioning to you before of around 100 motor neurons decoded from the forearm muscles in certain digit tasks. So as I was saying before, when you look at something like this, you have now it looks just like a color barcode, but if you go deep in that, you have the last neural coding of that specific task. So that means that if you have a certain contraction of a certain digit, then you can look at what are the neural mechanisms that allowed for that specific control. And you can do that in a very precise way. And so these are other examples, just to give you a bit of a zoomed version of those barcodes. You have different finger control, and here you have different lines. Each line is a motor neuron, and each line is made of a number of vertical lines that are the exact instance of activation of each motor neuron into the spinal cord, decoding those findings. Now, the fascinating thing, which I will not discuss today, maybe I will, maybe, is that when you decode the motor neuron activity in the way that I've shown, because you have the output of the system, you remember this is the output of the full system, then you can understand something about the input that is needed to produce that output. And so you can go backwards, up to spinal circuits and even up to the brain. And indeed, as I said, I will not touch these topics today. You will be able to read the brain oscillations directly from this output. And if you are interested in that, just a few days ago, Jaime and others published this nice perspective paper in Nature Biomedical Engineering discussing how that is possible and why, from peripheral interfacing, you can actually read the brain, which is quite, in my opinion, exciting, because that means that you can put a bracelet with a few electrodes in your wrist and you can read brain oscillations. And that is indeed possible, but I will not discuss much on that. Okay, this was a bit of the didactic part on these methods. As I said, I've discussed this method many times. Maybe it has been a bit boring for some of you, but I hope useful for some others. And now I will go to a few applications, and some of these applications are quite new, so I hope they will be new for at least the majority of you. So this is just a screenshot on the number of fields in which our group and others have applied these techniques. I will not discuss all of them. Here is an example of non-human primate recordings from both the brain and the muscles. And remember, these techniques can be applied also for intercortical recordings. These are examples of recording from infants. This is spinal cord injury, and this is bionics. I will touch on some of those, especially on the newest results, and I will start with the basic motor control. So behind all these applications, there are also studies on the basic motor control. And I start with the basic motor control because this will have an impact on some of the examples that we'll do on applications mainly in bionics. So let's start from the basic motor control, and let's ask a simple question. We know that we have a certain number of spinal motor neurons. We have learned it half an hour ago. Now we can ask ourselves, can the central nervous system, or does the central nervous system control every motor neuron independently in the spinal cord? So you may think that it's in principle possible the nervous system address each of those motor neurons independently and can control them independently. So again, because this is a summer school, this is Basmajian. Basmajian, one of the pioneers on biofeedback techniques, and mentor of Carlo De Luca, who we have seen before. And in '63 in Science, it's only a 2-page paper, this one, so you can read it quickly if you are interested. It's a nice paper. Made a very simple experiment that was revolutionary at the time. He recorded from a muscle, as you can see, which is a muscle from the end with a wire electrode. Remember, Adrian had proposed the concentric needle already in 29, in the 30s. So this is 30 years later, there were developments in these techniques. And it's a wire electrode. It's not anymore a concentric needle. It's an electrode that is inserted with the needle, but then it stays in the muscle without producing pain or being unpleasant. And this is an oscilloscope, and directly from the oscilloscope, so without any decoding, he was asking the volunteers to have a very, very low level contraction, low force contractions. And if you decrease that, you can do this experiment yourself. If you decrease the contraction at a certain low, very low level, this would be 1 or 2% of your maximum force, then you start seeing individual spikes that are repeating over time, and just by naked eye, you can recognize that the spikes are repeated. So those are obviously spikes from a group of fibers innervated by a single motor nerve. And Emma's mission asked to these healthy individuals to increase or decrease the distance between consecutive action potentials. So what does that mean? Well, that means that he asked basically to modulate the firing frequency of those action potentials, so the firing frequency of the motor neurons. And the question was whether or not the central nervous system could control motor neurons individually. Actually, this is an important study because it's the very first proof in humans that humans can control single neural cells with biofeedback volitionally. I think a decade later, this was demonstrated for cortical neurons, but this was first demonstrated for spinal motor neurons because in spinal motor neurons, it's so much easier to record from. So there's the first paper in which the first result is that the humans can control single motor neurons. So if you ask humans to do that, indeed they can do that. You can try yourself. So you can decrease the interval between two spikes. That means that you increase the frequency, or you can increase the interval. That means that you decrease the frequency. If you are given a proper biofeedback or even a very simple biofeedback like this one, you can do that task in a perfect way. You don't need training. You do it immediately. All right, so from this study, there have been some interpretation, not much at the time, a bit more recently as well, that maybe this means that indeed the central nervous system can address every neuron, right? So if you switch to another neuron, indeed the central nervous system can control this one and so on. So plasmation could switch among different neurons and they could be controlled. So that's interesting because even if there are not that many, we've seen that there are tens of thousands, and to think that every single one is driven independently, that is could be surprising to some, at least from the computational point of view side. All right, this is only part of the story though, because a long time later, so this is 60 years later, this Mario, who is here, is there. This Mario Bracklin was a PhD in our group until 22, so some years ago, had the idea to repeat those experiments. Those experiments were 60-year-olds, and everybody could do those experiments in any lab. In any lab, you could put a needle and see a motor neuron activated. But Mario had the brilliant idea of saying, well, let's now control two motor units of motor neurons at the same time. So let's put the firing of a motor neuron, let's say number 2, on one axis, the firing of motor neuron number 1 on the other axis. And then let's ask the subject to go with a cursor over the full 2D space. So what does that mean if you can do that? If you can do that, it means that indeed you are controlling those two motor neurons completely independently because you can go in any part of those space. You can switch on one and switch off the other and vice versa, or you can modulate the two firing by having one increasing the firing, the other decreasing the firing, and so on. Okay? And he had people training for 2 weeks on this task. So it was quite boring for the people participating, but for 2 weeks they tried to do this. So every day they were selecting with the techniques I've indicated to you before. So you have muscle recording, decomposition in motor units. They were selecting random motor units, putting it on these and asking to do this task, reaching this type of targets. And then in the same session, of the 2 units, they could be close in terms of recruitment threshold, they could be far away in terms of recruitment threshold. They tried a number of combinations and they repeated this for 2 weeks. So to cut a long story short, none of these subjects could do that task. They couldn't explore the full 2D space. What they could do easily was to go in the diagonal. What does it mean that they go in the diagonal? That they are controlling these 2 motor neurons in a fully correlated way, right? You increase the firing of one, increase the other, and vice versa. You decrease the firing and so on. So the main behavior was going to the diagonal. It was very difficult to break this behavior. It was very difficult to do something else than this. So what does that mean? That yes, you can control single motor neurons, but then when you start to try to control them independently, you cannot do that. So as always, again, because this is a school, you have to know that there is never new research. There is always old research that is revised. And indeed, also in this case, that was not a huge surprise, or should have not been a huge surprise, because already from the 70s, maybe before, but I think this is one of the first papers done properly on this phenomenon. Already from the 70s, we knew that if you record from 2 motor neurons, at that time they could record only from 2 at a time, and these were wire recordings. Then they were decomposed, and if you made the cross histogram between these 2 firings, you get something like this. You always get a peak close to 0. What does that peak mean? That the 2 activities are correlated. And this historically has been called short-term synchronization. Short-term because it's close to 0, right? So that means that there is a likelihood above chance that those neurons are firing at the same instant of time or very close to. Okay? So this was already telling us that there has to be a sort of correlation between neurons. And the basic concept seen in a bit more modern way is that motor neurons receive some sort of common input. We have to define what it means, that impose correlation in their activities. And so if you want to make a simple model of control of motor neurons, you have a common input that branches to many motor neurons. Now you have 3, but you can imagine a group of motor neurons, a certain group. And this motor neuron will also have some independent inputs, but the main drive is the common input, and therefore there is some level of correlation in their discharge outputs. Okay? I say some level of correlation and not perfect correlation, even if you had only common input, because those motor neurons are obviously nonlinear. So if you have even a perfect common input, because of nonlinearity and because of different intrinsic properties, the output is not necessarily perfect correlated. But even within the nonlinearity, you have some correlation, which is reflected in the studies I've shown you before. Now, our idea is that in recent years is that the control of movement occurs by clusters of these motor neurons. So the central nervous system sends a certain number of common inputs, different commands to different clusters of this motor neuron. Each cluster of these motor neurons connect to muscle fibers, and therefore it has a certain functional output. And by combining these clusters of motor neurons in various ways, you can determine behavior. So can you demonstrate this experimentally? Can you demonstrate that there is a common input in groups of motor neurons experimentally? Well, in principle, yes, because remember, let's say that you have a group of motor neurons, let's say that you have a certain number of inputs projecting common into a group of neurons, but remember from the output, you can try to infer the input. For example, because common input impose correlation, you can compute the covariance matrix of the activity of those motor neurons, and then you can build a graph in which each motor neuron is a little sphere, and you put motor neurons close by if they are mostly correlated. If you do something like this, this is a simulation. In a case in which you have two input, you will see that there are two clusters that you can identify with the clustering algorithm, and then you have identified the dimensionality of the input. And you've also identified which neurons receive which input. Okay? So experimentally, you see exactly this. So those were simulations. Experimentally, these are data during not exactly a cycling task, but a cycling which is blocked at a certain angle. And these are recordings from the legs. And these are the motor neuron graphs that you see for that specific task. Now you can see that you have two clusters in this way, in this case, another two in this other. Actually, these are the same, but with some slight difference, which I'll not discuss now. In color, you have the different muscles that these motor neurons innervate. You see that the clusters are not corresponding to muscle. You don't have a cluster for each muscle. You have a macro cluster, for example, that involve a number of muscles together. So that means that all the motor neurons innervating all those muscles are receiving the same common input. So muscles are, in our view, irrelevant for the nervous system. The nervous system only see those, for example, in moving my arm, those 20,000 motor neurons. It has to activate those 20,000 motor neurons in a certain way. It takes those 20,000 motor neurons, make clusters. Every cluster has a functional output, and through a reward base, organize in the best way this clustering. Irrelevant from the muscle part. At least that's our view. And this is an example for other participants of others of this class. So you can see here you can exactly identify the different common inputs that the motor neurons receive. So we call these common inputs motor neuron synergies because, and this is a perspective paper of a couple of years ago where you can see all the evidence for this view, which is still just a theory, can be disproved with other experiments. We call these motor neuron synergies because classically, you talk about synergy, we're talking about muscles. So there have been for a long time a theory of muscle synergy. There's still a theory of muscle synergy, that is, the muscles are controlling coordination. We believe that you have to go at the level of motor neurons to really find the synergies. And the synergy will be defined at the motor neuron level as clusters of motor neurons that are projected to any muscle. That doesn't matter. Depending on which muscle or which portion of muscle they are projected, or which multiple muscle they are projected, they will have a certain motor output that could be a sort of a motor module. And then you can mix this motor module activating many clusters to determine behavior. So these are a sort of the assumptions that we wrote in this review paper that are basically what I explained to you. The muscle anatomical borders in this view are irrelevant. You only look at motor neurons. Interesting, this theory implies that you explain dimensionality reduction, interestingly, because the nervous system doesn't have to provide a signal for each motor neuron, but it has only to provide a signal for each cluster of motor neurons, in this case 3. So you decrease the dimensionality dramatically, that explains the computational load. And at the same time, you are extremely flexible because in principle, you can change those clusters and you can readapt your system, for example, to learn completely new tasks. Now, we don't know at which point this flexibility extends, I have to say. We have still to do experiments on that. But there is certainly a compromise between the need to have some of these motor modules firmly fixed, and the need, of course, to be flexible and to learn new tasks, or to adapt or to improve on all the tasks. So it's interesting to apply this technique in pathological conditions because you can see things that are much clearer than from the muscle perspective. So these, I think, are beautiful data. They are from the Shirley Ryan Ability Lab, the group of Jose Pons. And this is quite standing. So the person is just standing, and signals are recorded from muscles of the legs. And this is the clustering of motor neurons from all these muscles. And you see the color code when you're quite standing. So you see how beautiful you have two points, two clusters. And if you look at the neural signals that they receive, you really have a state machine that goes like this, from one to another, from one to another. What is that? That's the inverted pendulum, right? So we are an inverted pendulum with microforce forward, microforce backward. Neural is exactly this. These are how the muscles counteract the microforce. So that's a very nice neural overview of quite standing. And then, of course, you can go into this and you can look if the neural input are burst-like, for example, or are continuous but with different phases and so on, right? Things that we don't discuss now. But now this is interesting to see what happens in patients with stroke. So you see what happens with patients with strokes. That input is not common anymore. It's spread almost randomly in all these motoneurons. If you record for muscles, you don't have any view of this. You will just have the impression that there is some muscle activity. If you look at muscle synergies, most often you will conclude that the dimensionality is lower. The dimensionality is lower just because all muscles receive almost the same random signal. And of course, when you do the envelope, they are all similar. But it doesn't mean the dimensionality is lower. Actually, if you talk about dimensionality is paradoxically larger, but larger in a way that functionality is not positive. So you see the two type of views. Okay. So these were some examples of motor control studies with these techniques, and I realize it's already 1 hour that I talk. It should be quite heavy to follow for 2 hours. So we try to switch to topics that maybe are a bit more applied and sometimes more interesting because they are closer to patients. So I will now discuss with you, and you will see that there is a continuity between the topics. Some work done in so-called bionic medicine. So when you have to control a robotic arm and a patient with an amputation has somehow to send commands that has somehow to be decoded in order for that arm or hand to be controlled following the usual intent. Okay, so this is a classic problem, which is called myocontrol. Myo comes from muscle, control from control. So you use muscle signals for control. And it's called in this way because the classic solution is to put electrodes covering the stump above the amputation to record from the muscles that are remaining. And if you have some of these muscles related to the movement that you want to decode, you can then translate this muscle electrical activity into functions of that prosthetic hand. So as I said, this is called myocontrol, and these are techniques that have been investigated for really a long time. They are on the market, systems like this, and thousands of patients are using this type of simple control signals. So there is a paradox in myocontrol that is well known by industry. That is that the higher is the amputation level, the higher you go in amputation level, the less muscles you have from where you can pick into the signals, right? So if a patient has an amputation at the shoulder level, there is no muscle that has anything to do with the hand movement, and so there is no possibility of intuitive control. The subject can control it by learning another type of control, maybe, but also there is not many more muscle tissue actually to interfere. So this is a paradox because the higher is the amputation, of course, the more the degree of freedom to control, right? So when you need this control more, the control fails because of lack of remaining muscle tissue. So how can you solve this problem? Well, you can solve this problem in a few ways. At least two of them are very obvious. The most obvious is you say, well, that's fine. If I have an high-level amputation, I don't have muscle tissue, but still I have the nerves. I have the nerves that were controlling the end. I can put microelectrodes in those nerves. Even if I have an eye amputation, I can find the nerves responsible for those movements. I put electrodes and I decode the movement at that level. So this has been done. It's been done extensively, especially 20, maybe 30 years ago. And then people found out that decoding the motor efferent fibers from nerve recordings was very, very difficult. The signal to noise ratio is very poor. You have both efferent and afferent fibers that goes into directions. The electrodes have to be inserted into nerve tissue so that you can have damage and so on and so on and so on. So it can be done in principle. In practice, we don't have good enough electrodes, microelectrodes for nerves at the moment to solve this problem. Maybe we will have them in the future. For sure, we'll have them in the future. At the moment, there is no clinical application with nerve implanted electrodes to decode motor intent in patients who want to control a robotic hand. On the contrary, there are clinical applications that use another trick, and they use the trick that leverage my statement at the beginning. You remember at the beginning I said that muscle fibers are biological amplifiers for motor neuron activity. Well, in this case, you don't have anymore that connection, but you can create it surgically. So you can take the nerves at the stump, instead of inserting electrodes in the nerves, you can surgically take the nerves and bring them up. And what happens if you bring them up? They will reinnervate the muscle tissue next to them. They will do that. The nerve is eager to innervate the muscle tissue. Will go there, innervate that. And then what have you created? You have created an amplification of the nerve activity, and then you can record from muscles. And then muscle recording now will not anymore reflect the normal activity of this specific muscle, for example, the pectoralis, but it will reflect the activity of the nerve that you have moved in those locations. Okay, so this surgery is called targeted muscle reinnervation. It was proposed by Todd Kuiken at the Rehabilitation Institute of Chicago 25, 30 years ago. These are some of the papers. In particular, this is a famous one, but the technique was published a few years before that, so it's at least 25 years ago. And it was demonstrated in humans already at the time, and now there are a very large number of patients who have this surgery, and then they have interfaces that are as simple as these ones, and they may have reinnervation in the chest, in the back, on the shoulder, depending how high is their amputation. All right. So now to make you understanding how this works, this is a video from when I was in Germany long ago from the Göttingen University Hospital in our labs at the time. This is a patient with an high-level amputation. You see these colored maps. These are just grids of EMG. The colors indicate the electric field, which is EMG activity. And this patient had this reinnervation in all these points where you see the grids around 6 months before this video was taken. So the patient is doing the task that you see at the top in the title. He's really doing this task. So the tasks are done by the brain. They go down to the spinal cord. They go to the motor neurons. The motor neuron discharge. With this amputation, they would stop at the stump, but now they are redirected to go in other muscles. And so we can read from them. So because we can read from them, we can decode all those tasks, right? And some of those tasks are very distant tasks. For example, end tasks. We can do that because we are reading from the nerves. We're not anymore reading from the functional point of view from the target muscle. The target muscle are used as screens, as amplifiers. They're not used anymore for their function. Okay? All right. So this is very well established. It is done in several clinics for bionic reconstruction or for bionic substitution. There is though a limit, and the limit is the following. Let's say that we take a nerve that is cut at the stump, this one. Let's say that this nerve, doesn't matter which nerve it is controlling the end, and I want to have this information, so I want to read from this nerve. So that's fine. I move this nerve, as you can see, to this muscle. Now is the blue muscle. Now this nerve goes to this muscle, and I can read from this muscle. That's the procedure we just discussed, right? Now, the problem is that the nerve that we are redirecting very often is a polyfunctional nerve. That means that there are a lot of functions embedded in that nerve. The nerve can provide, for example, the movement of the small digit, at the same time also the movement of the middle finger, or it may have controlling the plantar movement of the hand or whatever. So there are a lot of tasks embedded in this nerve. In classic targeted muscle innervation, I take the EMG signal from this muscle, as you have seen before, and I use that from one command, one single command. Doesn't matter that there are many tasks decoded there. I use one command, and so for every degree of freedom, normally you have to have two reinnervation points. Two reinnervation points, because if you consider, for example, elbow flexion and extension, this is 1 degree of freedom, but you have one direction for flexion and one direction of extension. That's two signals, there are two reinnervations. So how do we get more signals? How do we get more signals from this operation? How can we get more signals from this reinnervation? Well, some of you may think of what we have said in the past hour, and maybe you can get some ideas. We will discuss these ideas in a moment. But we can start from pushing even more the boundary of surgery. So this is work of Paul Sederna in Michigan. And the idea here is fully surgical. You say, well, this nerve is a polyfunctional nerve. I want more function from this nerve. Surgically, I split it. Instead of taking the full nerve and going in a muscle, I split the nerve, and then I take a muscle graft and I put around each of these nerve branches. So I don't know exactly the nerve branches, what they are coding, but I hope that they will have slightly different functions, or at least that the majority of them will have a slightly different function. Okay? Then I can place electrodes. Here they can even be implanted electrodes, but in the muscle tissue. So now you see that this is an exploitation of the idea of using muscle tissue as an amplifier. There's a strain because you actually harvest, normally you take this muscle tissue, for example, from the leg, you harvest muscle tissue, which is an amplifier, and you put it next to the nerve that you want to amplify. So this works. This has passed the preclinical study level, and it is in humans. So this is a human surgery. Yeah, I apologize for the bit of the gory images, but I think this figure, they will be the only one. And I think this figure is showing very well what is done practically. This is exactly as the skin that you have seen before. So here you have a nerve. That doesn't matter which nerve this is. This is taken from a paper 5 years ago in Science Translational Medicine, where this is the original paper for this technique. This is split, you see, in 3 parts. That's surgically split with a surgical blade. They don't know how they split it, right? I mean, you split as you can. And then these are muscle grafts. You take some muscle tissue somewhere. And then here it's a bit less clear, but each of these muscle grafts is sutured around these three nerve branches. All right? Then in each of these, you put an invasive electrode. At the moment, in these patients, the invasive electrode goes out through the skin, and it goes to an amplifier, and you can now have from this single nerve, you have 3 commands. And here are, from the original paper, the variations of this type of operation that you can do, either in the upper limb or even in the lower limb. Okay. So that's fascinating. That's great. That is applied in clinics. The idea is brilliant. Of course, it comes from people with a surgical background. It is possible. It is not such an extreme surgery with respect to the reinnervation. So it can be done clinically, and it is done clinically. You have some problems in implanting the electrodes with a fully wireless system, which is not yet done yet. But patients that have this type of intervention, they have very nice control capacities. So this has been done probably in less than 10 patients. It's still only one group, although there will be one of these surgeries in Europe before the end of the year in Vienna, but it's still quite limited. All right, what else can you do instead of doing all this? Well, from all I have told you, so the splitting of this nerve is just because you want to identify different nerve fibers, and then you put different muscle tissue. But you don't need to split it. I mean, here you could have a single muscle tissue, and in principle, for the techniques that we have discussed, you can decompose the motor neurons, and then you can identify the motor neurons that are responsible for the different tasks, right? For example, the motor neuron responsible for the different tasks will have correlated activity. So you can use techniques that we have seen from the motor control studies to group those motor neurons in a blind way. You don't need to know that, and you can separate the activity. So what is this? This is a mathematical surgery, splitting the nerve instead of a real surgery. You take the full signal and you divide it. And this works particularly well if you use a microelectrode array in the reinnervated muscle, because the microelectrode array is very selective, provide you a lot of signals, and because in the future this is not that electrode array implanted, but this is another type of system. But just to show you, in the future you can also have a full implant of microarrays in the innervation side, decoding the motor neurons and going back. So this concept was done, this is work, Medical University of Vienna, also with our group some years ago. That's a preclinical study showing that concept. So this is a rat, as you can see. The nerve innervating the paw of this rat was transferring one of the head of the biceps. And now the idea was to cover this, was to understand the neural activity there and separating the different functions. So here you have microarrays, you can see them in this photo, microelectrode arrays, identical to what we use in humans, that are recording from this muscle. And you get this type of recordings. And then with the decoding I've shown you before, with this kind of recording, which is this observation, you go back to the motor neurons. And from the motor neurons, you go back to the individual tasks. And in this preclinical study, everything worked well. But of course, the animal is anesthetized. You have a lot of limitations. So now, more recently, this is work of Laura, actually, that just been accepted in Nature Biomedical Engineering, so we're very happy for that, just a few days ago. You can translate this concept to humans, and that becomes really very interesting, because you have this polyfunctional nerve. You see here all these colors are different functions. This is sent to a muscle. That is what targeted muscle innervation would do. And you have all this mass of activity, you see. Some fibers are innervated by neurons corresponding to this cluster, some other to this, and so on. And so here you have a very complex activity. In classic TMR, you use this as a single signal, which is the sum of everything. That's enough to make one control. But then what we did was to put the microelectrode arrays inside and then separate each of these, not only, but also go back to the individual clusters. What are these clusters? That are the cluster of motor neurons I was mentioning before when talking about the theory of motor neuron synergies, right? It is exactly the same. So the idea now is that out of these signal, now you have a number of action potentials for the different motor neurons. This is a patient in our labs. Of course, the preparation is quite messy because it's in the lab. You have the implants here. This is the connector. You probably don't see the filament, but this little dot is the insertion point, and we have more than one implant. And the result that you will see are from 3 patients. Here, just for your curiosity, are the nerves that were reinnervated in the point where we put these microelectrode arrays. So these are the typical recordings from these patients. They are very nice. Here you have channels of the microelectrode array from one muscle, and with the different colors and numbers, you have the motor neuron, the different motor neurons. You see, for example, this is one motor neuron, and then number 1 will be another one, and so on. All right. You can also look at the territory of those motor neurons in the muscle. Because you have the microelectrode array, you can look where the action potential are distributed. And these are the territory of different motor neurons. So this is an indication of the spread of the reinnervation. These are action potential of single motor neuron. This is the distribution of spiking of different motor neurons. This is a raster plot of the motor neurons, and so on. Not only, but then you can count how many motor units are active in each task or are shared between tasks. So you can start to make a preliminary analysis of how to group these motor units. So these are the different muscles that have been reinnervated. These are the different tasks. and in color you have how many units are shared among these tasks, all the others being unique for that task. But then you can make the final step, which is obvious at this point, and you say, well, we know what is the theoretical governing principle for those motor neurons, right? We know that, as we have seen before, the central nervous system, at least in our view, will cluster motor neurons, and then these will go in a certain polyfunctional nerve in clusters. They reinnervate, there is all these, we can decode, and after decoding, we can then, in principle, we can then recluster them and extract the common input to each cluster, and each common input is a signal of control. It's what by surgery you will obtain if you split the nerve, and if you're lucky enough to get the right clusters, though. In this case, you always get the right clusters, right? So that's really a mathematical surgery, all done through decoding and blind reclustering. So can this be done? Well, if you take all the neurons decoded from these tasks and you apply a factorization analysis that implies to look at the correlation between them, you indeed find different clusters that corresponds to the distribution of the different motor units to explain different tasks. So in this case, for example, you have 6 groups. 6 groups, they are blindly identified. This 6 group of units explain 6 different tasks on that polyfunctional nerve. So this is equivalent to a split the nerve in 6 parts, and having been lucky enough to get the 6 compartments that are corresponding to these actions. All right? Then, of course, as I said, once you have done that, now in color there are the clusters. Here in the signals in time domain are the common input to those clusters. So here a contraction that goes up and down, up and down, up and down for each command, and so for each task. And then, as I said, obviously then you can use these common inputs for commands to a prosthesis. Okay? So does this, we use just 10 more minutes, then we stop. I'm getting time myself. So, so does this work also when you really fully translate? So first, these are with implant electrodes and so on. Laura arrived at this point, and now in these weeks, we are doing the online control of the prosthesis. This is very complicated. You can talk to Laura about that. So it will not be something that we will have in a few days. But we can put the same concept and have a look at the translational activity with full command with another interesting piece to the puzzle. And the piece is the following. Each of these clusters corresponds to a certain task, but it's not exactly a task. Remember, each of these is a motor module. It's not necessarily a task of a single muscle. It's a coordinated task that makes a motor module. It's what we call the synergies, right? So the combination of these synergies provide a complex space of tasks. So this happens also from the biomechanical point of view. If you look, if you study my hand and feel my hand during the entire day, and then at the end of the day you take the kinematics, which is 25 degrees of freedom plus, and you put it in a dimensionality reduction algorithm, you will find out that you have much smaller dimensionality than 20 plus degrees of freedom, right? Because the degrees of freedom are correlated, and most of the time we do actions in which we have coordinated and correlated activity of degrees of freedom, right? So the synergy idea is also at the kinematic level, and one can ask, well, would neural synergies correspond to synergy at the kinematic level? In principle, they have to correspond, right? Because the neural synergy determine a certain task. But if they correspond to the kinematic level, can we design a robotic hand in a way to embed a synergistic type of control? Or better, embed a mechanical design that is synergistic? And it can be done. So this is another recent in which the idea was to merge human synergies at the neuron level with robotic synergy, meaning building a robotic part with mechanical synergies. And somehow this is a sort of the arrival point, at least for what we have now. You will see in the next slides of what at the beginning I posed you as a problem of neuromechanics, right? So now we are really trying to merge neuromechanics, even from the mechanical design point of view. So this is a synergistic prosthetic hand by Antonio Bicchi, with whom we had the pleasure to collaborate. If you look, if you have looked at the video, it was very short, but anyway, this one, this is a motor that makes this type of task. The two index and middle are closing down. And then you have another motor that does these other type of tasks where you have this kind of operation, right? Now, the two motors can be linearly combined. If you linearly combine the two motors, you can give infinite linear combinations to the two motors, obviously, and you create a two-dimensional space of mechanical posture of the hand. For example, you can have the full closed hand, that's obvious. If we have the same weight of these two, the hand will close. But if you have intermediate, this of course is a real hand, but if you have intermediate, you can even do tasks like this. As you maybe, I don't know, if I have an example of this with a prosthetic hand. But the idea is that now you have two synergies, and with these two synergies, you are able to build an infinite two-dimensional space of hand postures. Okay? And what we did with Antonio Bicchi, this is also recent, was the following. Let's try to extract the motoneuron synergies in terms of common input. In this case, it was two-dimensional, and associate these to the two motor, meaning match these with these two synergistic tasks. And this works. This is a video with the end on the desk. There is an healthy person. This is, for example, the first synergy. This is the end, by the way. So this is the first synergy. You see, the end is soft, and it is these are both synergy. You see that it seems quite a natural end, right? It is only two motors. And I don't know if the video stopped. No, there is still. Oops, it stopped before. Anyway, and still the other synergy. So you see that the movement of the end is quite natural. It is natural because it is a soft end. It's natural because it combines the synergies, and therefore, instead of seeing predetermined task, for example, if you have a classic commercial hand, you have open and close and rotation, and you see these are robotic tasks. I mean, opening. Well, in this case, it seems almost a human task because you have an infinite space of adaptation of this posture, and this is. I have better. There are better videos in the publication that was cited in a few slides ago, so you can download the videos. I only have this whole video. This is a patient. She has a congenital amputation, and for the first time, she's trying to play with the hand. I don't want to demonstrate or state anything with this video, but just the natural type of movement that this hand is doing. So she is training her motor neurons to control the two synergies in order to produce some specific task. As I said, if you look at the paper, there are a number of videos that are much better than this one. All right, now 3-4 more minutes. We say that when you have this polyfunctional nerve that is going to a muscle, you can separate it, right? But then why to stop at that? You can also say, why don't we take all the nerves that are innervating the arm and put them in the same muscle? And then we will have a bioconnector. We only have one implant, and we separate it in the same way. So this is also something that we are trying to do. This is a preclinical trial already from 21. So this is Matthias Luft in Vienna. And this is a rat model in which you can see that there are two nerves in the same muscle. And these two nerves are indicated here in the same muscle. And this was a proof of concept study where there is the possibility to record EMG signals and also the possibility of separating the motor neuron from the two different nerves, but also separating motor neuron within the same nerves for the different functions. So I will stop here for this part and conclude in a moment by saying that in principle you could have in a single muscle or even muscle graft, you can direct the full neural drive for the full arm. If we talk about an arm, you can decode the motor neurons of all the nerves. You can even separate the compartments in the nerves, in principle at least. You can derive as many commands as you need, or in principle, as many natural commands as there are, and control a system that is as complex as needed, as complex as the biological arm. All right. Now we are light years from this vision, admittedly, but the components are there. The scientific components are there. It is something possible. It's just very difficult to implement. I will close by saying that this type of technique, since this is an application part, this type of techniques in two final slides have also the possibility to be used for non-medical applications. So you can, for example, have a wearable technology that is now becoming a neural interface because you can decode the motor neuron or even decode the upper neurons, for example, for controlling virtual reality. This is an old slide of Control Labs. At the time, this was an independent company. Now it has been merged by Meta. And this is the time they were very happy. This would have been almost 10 years ago. They were no, well, 7, 8 years ago. They were very happy because for the first time they saw for this bracelet this spike in activity, which are indeed single motor neurons. And in that case, they've just been very lucky, having a very skinny subject for which without any processing, you could still see the spikes. But even if you are not in those lucky situations, even if you cannot see the spikes, as I've explained to you, you can take multi-channel signal from a bracelet of this type and decode in various ways that activity. And this is just to finish work of Irene Mendez, also PhD in this some years ago, decoding single finger control by decoding motor neuron activity from a system that is mounted at the wrist. So in principle, a wearable system that could be associated to a smartwatch or similar. All right. I thank you for the attention and for the patience. So it's 1 hour and a half. Listening is quite tough. Thank you.